So I'm Meredith Pochardt, and I'm going to just piggyback off of Michael's talk, because you definitely can't have enough Eulachon talks, or Eulachon. And so I've been working with Eulachon for quite a while now, and I live in Haines, Alaska, on the traditional homelands of the Tlingit people, and that's where a lot of my work happens in Alaska. But I'm also part of the Karut program, working on the Haisla territory as well. So Michael gave a great introduction into Eulachon. I'll probably say Eulachon or Eulachon or, hooligan. It's all the same fish. But so we'll go over a little bit of background on them, but then we'll kind of get into more of this broader regional monitoring and some of the abundance estimates that we're developing, and then kind of just wrap up real quick. So yeah, this is just another beautiful fish, or photo of Eulachon. So as Michael touched on, there's a long history of the cultural connections with Eulachon. So there's a whole network of grease trails throughout the Pacific coast, and these are trade trails from the coast to interior. This is what made a lot of the coastal native groups, or First Nations groups, really powerful. It's having this Eulachon grease. And so this is up in Haines. This is Dwayne Wilson making a batch of Eulachon grease. This is the big smile that you get when you have a jar of Eulachon grease. And then, obviously, a smokehouse full of Eulachon is pretty amazing as well. So this doesn't really have anything to do about eDNA, though, so we'll get into that. As Michael mentioned, Eulachon populations basically south of the Nass River have been declining since the 1990s. They're also listed as threatened in the southern part of their range in Washington, Oregon, and California, and in central BC, in southern BC. So, yeah, we've been working on Eulachon monitoring in southeast Alaska and central BC for quite a while, using eDNA. So in northern southeast Alaska, in the Haines area, which is up there north of the Inside Passage, we started monitoring in 2014 using eDNA. And prior to that, we were using just mark recapture methods. And then central BC folks, I believe we began in 2019, but it could have been a little bit earlier than that as well. And then some other rivers in southern and southeast Alaska we started monitoring in 22, and then Yakutat in just this past spring. So we have this problem. We have this known declining population, and we have kind of a lack of hard data to support these declines. And so our big question is, can we use environmental DNA in a quantitative or a management relevant way to monitor these populations? So this is where eDNA comes in. I don't have to explain that to you guys, so it's great, you guys all know about it. And I think Michael maybe had the same photo. So these are kind of, for the eDNA components of the Karut project, these are some of the big things that we're trying to tackle: is spatial variation over 12 rivers, a time series at the Camano River going back from 2019, and then also comparing those with the larval outmigration and egg drift data, and then some new marine larval monitoring. So I'm just going to fly through some of these graphs. We can just look at trends and see how things compare, the eDNA signal, and this is the egg count. This is also in 21, and the Camano River, same trends. We're seeing that there's a pretty tight correlation between our eDNA concentration and the egg count. And then also in 22, we're seeing similar things. So how does that compare with larvae? Sometimes it's pretty close, sometimes it's a little more hit and miss so far. So this is the Kildala River. It's another central BC river. And then a couple of years of that data. And then Camano. We are seeing similar trends where things are tracking, but not quite as tight as that egg drift data is. And then, so just to touch, so all of that and the Karut project, we're using qPCR, we're using the assays that Michael's developed. In Alaska, we started using ddPCR, or digital droplet PCR. It's just part of how that program got going. We have a partnership with Oregon State University to run those samples. I won't go, you guys probably know a bit more about ddPCR than most people I talk to, but basically you partition that sample up into 20,000 droplets, and if you have positive DNA signal within that droplet, it will fluoresce as positive. And so how are we using that for abundance? This is the positive control tissue sample. Obviously see lots of fluorescence there. When they're really rare in the system, we're seeing still some positive fluorescence, but much rarer. And then when they're really abundant, you kind of see this rain that comes down. And so we compared that with our mark recapture methods. on the Chilkoot River, and we're looking at two different kind of metrics of this. One's the size of the peak of the eDNA concentration, and the other one is that the area under the curve. So when we have multimodal runs, the area under the curve can be really important to get that full kind of overall abundance estimate. I'm just gonna fly through these last ones. This is binomial Poisson regression comparing that, the eDNA concentration, size of peak, and area under the curve with the marker capture data. We're seeing a pretty tight relationship there. And just another cool thing is just comparing eDNA with wildlife observations during these fish runs. As Michael mentioned, it's a huge congregation. It's like the first thing that comes back in the spring. And I'm gonna get out of the way. Yeah, this is, eDNA and sampling at all in remote locations is challenging. But this has been proven to be successful in developing a time series of data for data poor and indigenously important species. Okay, I'll get out of the way, and thanks to everyone.